Okay. All right, I guess we'll get started. Welcome everybody to the first path rounds of the new academic year. It's my pleasure to introduce our speaker today, Dr. Katey Enfield. For those of you who don't know, Katey is a new assistant professor in our department who just started this week, um, and is also a scientist in the Department of Integrative Oncology at the BC Cancer Research Institute. Dr. Enfield received her PhD from UBC under the supervision of Juan Lam, where she employed a multi-omics approach to discover and characterize oncogenes and tumor suppressor genes. While at BC Cancer for Graduate Studies, she was the recipient of a CIHR post graduate student scholarship and was the recipient of the Lord Skarsgård Research Excellence Prize, which is our BC Cancer Institute's award for top graduate student. Dr. Enfield, after receiving her PhD, undertook postdoctoral fellowship at the Crick Institute in London with Professor Charles Swanton, where she was studying the reciprocal relationship between cancer intrinsic genomic alterations and the immune response using the TracerX project. Dr. Enfield developed expertise in highly multiplex approaches for the spatial profiling of the tumor microenvironment and received another prestigious Marie Curie fellowship during this time. She is now continuing her research with our Prolong program at BC Cancer, and she's going to focus on spatial biology, spatial cancer biology, and B cell responses, which is what she's going to talk about today. So with that, we'll turn it over to Katey and look forward to her talk.
Thanks for the introduction, Will. Hello, everyone. And yeah, because it's kind of my introductory talk, I thought I would mention, kind of go through the different lines of research that I have followed throughout my academic career, which has been focused on lung cancer research. So as Will mentioned, I did my PhD here in Vancouver, moved to London, and then I'm really excited to be back again with lots of familiar faces and ready to continue to pursue all these questions that have come up over my academic career. But I also wanted to kind of paint a picture of how side projects can really change the direction of your research in exciting ways. So I would say that the focus of my PhD really was lung cancer genomics, but I did have a side project kind of originating after immunotherapy was introduced for lung cancer patients, working with Callum and Martial, really trying to understand how the spatial organization of cancer and immune cells in lung cancer sections might be maybe prognostic or important or relevant to patient outcome and immunotherapy response. And this really launched me into my postdoc, where I was kind of leading this development of the spatial immune atlas in the TracerX lung cancer study. But I also had an interest in B cell responses. And this really was another side project that I think is kind of blown into quite a research interest of mine. And I think the combination of all these things is what I'd like to focus on as a group leader here. So I'll just go through kind of the highlights of these different projects. So starting back at my PhD, working with different people at BC Cancer, VGH, and the DLE, we had this project looking at using multicolored IHC on tissue sections from about 20 patients with lung adenocarcinoma, so the major subtype of non-small cell lung cancer. And we were able to use these cool tools developed by Callum and Martial in their labs, where we did a hyperspectral image capture, spectral and mixing, cell segmentation, and applied different tools to basically call these different single cells into how they were stained, whether they were T cells, plasma cells, et cetera, and then run the cell sociology. So harnessing the power of spatial technologies and understanding how cells are related to each other in space. And the publication is just in the bottom corner. But basically, the main finding of this paper was that the frequency of CD8 T cells that neighbor tumor cells in these lung cancer sections was associated with relapse, even in this modest cohort of 20 patients. So this kind of sparked my interest in spatial cancer immunology and led to my postdoc. But just before I go on to that work, I'd also like to discuss how we took those same stains and the same cohort to ask a different question. So this paper was led by Kevin Ng, who eventually went on to do a PhD in London at the Crick with myself. So we were interested in these structures called tertiary lymphoid structures. And these can form in settings of chronic inflammation, including cancer and lung cancer. And we kind of observed them around the periphery of tumors. And we actually saw them in the majority of these 20 lung cancer patients' samples. And they're kind of formed of these different T cells and B cell zones. And there's a cell type called a T follicular helper cell, which is a CD4 T cell, which exists in the middle of these tertiary lymphoid structures, and essentially is there to train and select high affinity B cells that and support their maturation so that they can produce high affinity antibodies to something like a whatever target they're targeting. So the T follicular helper cells weren't as well kind of studied in the literature, and we looked at their levels in a cohort of lung adenocarcinoma patients that we had at BC Cancer. And we were able to show that their levels were increased in adenocarcinoma samples compared to paired normals. And this was kind of independent of stage. So this was a kind of consistent across different stages. And we also saw similar elevation of plasma cells, which is the type of B cell that eventually makes antibodies. Now, we were fortunate enough in this cohort to have paired genomic data. So we wanted to know whether what we were seeing under the microscope was associated with different genetic aberrations. And we did find that the levels of these TFH cells was correlated with tumor mutation burden. So more mutations, more potential neoantigens, and more TFH cells. And also a different type of antigen, a self-antigen that's aberrantly expressed in cancer, so cancer testis antigens. So these two types of targets were associated with TFH levels. So this is kind of interesting and suggests that TFH and TLS may be kind of directing tumor-specific immune responses. Now, while we were both in London, so myself in Professor Swanton's lab and Kevin Ng, my colleague, in George Katsiotis' lab, a slew of these papers came out that associated levels of B cells in some papers and tertiary lymphoid structures with immunotherapy response in various different cancer types, including non-small cell lung cancer. But these were all correlative studies, and we weren't sure of the mechanism behind this. So this was very, this kind of piqued our interest. And working together with Julian Downward's lab, who has lots of expertise in mouse models, another talented PhD student, Jesse, Kevin, and I kind of took this question and tried to pursue it using a combination of a new mouse model that they had developed in the Downward lab and various clinical cohorts of lung cancer. This mouse model is published. I have the citation in the bottom there, but essentially it's a KRAS P53 lung adenocarcinoma mouse model that's been a bit modified and then tested based on to see if it's more immunogenic. And we found that this mouse model was actually more responsive to immunotherapy. So we had a mouse model that we could investigate responses to immunotherapy, which was great. We had our 3 different clinical cohorts, and 2 of these received immune checkpoint inhibitor therapy. So we could kind of use a combination of mouse models and human data to ask various questions. Now, first I'll talk to you about the mouse results. And I have to say that this was led by Kevin and Jesse. I haven't done any mouse work, but I'll walk you through the results as best I can. So first of all, we wanted to know whether these TLS structures were observable in this mouse model. And we were excited to see these in the histology sections. And then we did a more sophisticated experiment where you can actually excise the lungs of these tumor-bearing mice and perform tissue clearing, and then do immunofluorescence staining to look at different cell types of interest. So here we're looking at T cells in green. So this is marked by CD3, B cells marked by B220, and our tumor-specific marker, TTF1. And when we look at the T cells, we can see that they kind of gather around the edge of the tumor nodules, but they also infiltrate a little bit into the tumor, kind of what we see in humans as well sometimes. And whereas the B cells kind of accumulate around the edge of these nodules. And using another technology, we could take a closer look and see that these B cell structures were sometimes forming tertiary lymphoid structures, which is what we wanted to see. We were excited to see these kinds of B cell zones and a T cell zone, which are characteristic of TLS. So next, we were interested in pursuing whether or not antibodies that were produced by B cells in this mouse model were detected in the serum. So we could see that mostly class-switched IgG and IgA levels increased over time in this mouse model. And then they did a cool experiment where they took the serum from a donor mouse and put it into a recipient mouse as a therapy. So essentially using the antibody repertoire from this mouse model as a potential therapy to check the effects on survival. And we were excited to see that in mice that received the sera, they had a benefit in survival, so they lived longer, suggesting that these antibodies were doing something to the anti-tumor immune response. Now, we were interested in learning more about what cell types could be contributing to this protective effect. We noticed that by flow cytometry, that the levels of NK cell fractions were elevated in the lungs of recipient mice that were treated with this serum. So they next performed an experiment where they blocked both anti-CD8 T cells, which are kind of the killing T cells, as well as the NK cells, and found that it was only the abolishment of the NK cells that actually influenced whether or not the serum had a protective effect in this model. So this was really interesting as well, kind of links that this antibody and NK cell response specifically to the survival benefit. And of course, we wanted to see the effect that we had when we introduced immune checkpoint blockade inhibitors, specifically anti-PD-L1 therapy in this mouse model. So again, we took the serum as a treatment and then also added this anti-PD-L1 therapy, and we saw an additive survival benefit. So this was really exciting as well, and suggests that it's the antibodies as well as this checkpoint blockade that have a combinatorial benefit to extending survival. Now, we were interested to know what these antibodies could be targeting. Are they targeting tumor-specific antigens, for example? So it's actually known in some of these mouse models that they can express this type of protein called a mouse endogenous retroviral protein. In mice, these can kind of be reactivated, and essentially they're remnants from evolution of very ancient retroviral infections that just kind of end up in the genome and are the so-called kind of junk DNA. But they can become reactivated in different settings, and we had characterized that there was one such mouse endogenous retroviral protein in the cell model, and there actually already existed a known antibody called 83A25 that we knew was specific to this target. And we could prove that this was relevant to our model when we kind of co-incubate the serum with this blocking antibody. It abolishes binding to the cell line, proving that it's targeting this endogenous retroviral protein. Now we can, excuse me, also use this antibody, this known anti-ERV antibody, as a therapy, and we saw similar survival benefit as we did with the serum. So this kind of was interesting that these retrovirus-specific antibodies were doing some, if not all of this benefit, survival benefit job. But we also wanted to take a more unbiased approach. So we did single-cell sequencing to sequence the B-cell receptors, CDR3 regions, to look at the different antibody clonotypes that we could see in our mouse model. And the predominant clonotype was this one we called J1KK. So it was synthesized And again, we use this ERV-specific antibody as a treatment, both the IgA and the IgG1 type of class switch antibodies. And we see the survival benefit a little bit more with the anti-IgG1 antibody. And as before, when we deplete the NK cells in this mouse model, we're getting rid of the survival benefit. Now, this is all very exciting, but we wanted to know, are we seeing anything similar in the human cohorts? So now I'll just introduce the TraceRx study, which I worked on with Professor Charles Swanton. This is a prospectively recruited clinical trial of patients with early stage resectable non-small cell lung cancer over 13 different hospital sites in the UK, and had a goal of recruiting over 800 patients. And the goal of the study is really to understand intratumor heterogeneity and tumor evolution. These patients undergo primary surgical resection with curative intent, and their tumor regions have multi-region sampling. And this is to enable these kind of this heterogeneity study. Patients are followed over time, so over adjuvant or no adjuvant treatment. And then unfortunately, a high proportion of patients do relapse. So we have relapse biopsies on treatment biopsies. And there is also another program led by Charlie and Miriam, which is an autopsy program called PEACE. But all of these samples undergo whole exome sequencing and RNA sequencing. And this enables us to kind of construct, based on the mutations of these multi-region sampled tumor regions, which mutations are clonal, so common to all different tumor regions, and subclonal, so common to just a subset of cells. So from this data set, the first question we asked was whether the expression, so the RNA-based expression of different B cell markers was correlated with disease-free survival in the two major subtypes of non-small cell lung cancer patients, so adenocarcinoma and squamous cell carcinoma. And it was interesting to see that there were histology-specific associations. So we did see that if you have higher expression of these B cell-related genes, this was only associated with improved DFS in adenocarcinoma patients. We were also fortunate enough with Professor George Cassiotis' expertise. This was Kevin Ng's supervisor. He has expertise in understanding endogenous retroviruses, not only in mice, but in humans. So we were able to interrogate endogenous retrovirus sequences that are known to code for proteins to investigate their expression at the RNA level in both the TraceRx cohort and also the TCGA cohort. And we actually detected expression of several in both adenocarcinoma and squamous cell carcinoma, but we did a bit more filtering based on whether or not they were expressed in normal lung, and we kind of narrowed on to this one endogenous retrovirus, ERV-K7. It's more highly expressed in adenocarcinoma tumor tissue compared to squamous cell carcinoma, and also against normal lung as well. So we actually, because we knew what the target was, we could run a test from the patient plasma, so kind of similar to the mouse serum experiments, taking patient plasma to look for look for the presence of antibodies specific to this target. So this is this HERV-K target. And we found that there were antibodies that target this endogenous retrovirus detected in the patient plasma, again, only in adenocarcinoma patients, but not in squamous cell carcinoma patients. So despite detectable RNA expression in both histological subtypes, we're really only seeing these antibodies come up in adenocarcinoma patients. And again, we were lucky to have paired genomics in the TraceRx cohort, so we could ask whether how these things were different correlated at the DNA, RNA, and protein level. So we found that the titers of the antibodies detected in patient plasma were positively correlated not only with the ERV-K7 RNA expression, so the transcript expression, but also with the gene copy number. And this ERV sequence is actually found within chromosome 1q, which is frequently gained and even amplified in the adenocarcinoma histology. So kind of provides an interesting scenario whereby a frequent genomic amplification event that happens in patients with lung adenocarcinoma is related to overexpression of this endogenous retrovirus, which after a certain expression level perhaps kind of breaks tolerance and then is recognized by the humoral immune system and antibodies. Now, we were curious to understand where these BCRs might be kind of maturing. So whether they're happening in lymph nodes, as you'd expect, or potentially in TLS, which are tumor adjacent and not in the lymph nodes. So we took this one case of interest, this patient CRUK35, and we could find they had quite a dominant clonotype, specific BCR encoding an antibody that was quite a dominant part of their repertoire. So we kind of investigated where we could see the earlier version of this sequence, the class switch version, and then the more somatically hypermutated, so advanced version of this antibody sequence. And if you just kind of focus on the magenta one, we don't really see this class switch somatically hypermutated sequence in the lymph node. We actually see it in 2 of the tumor regions. So this could suggest that this maturation is occurring beside the tumor in the tertiary lymphoid structure rather than lymph nodes. But it's just kind of a one-off example, and it's something I'm interested in pursuing in the future. Now, again, because in our mouse models, we were able to treat with immune checkpoint blockade and saw interesting things. Here we had in the TRACERx adenocarcinoma cohort, 7 patients that received immune checkpoint blockade. And here we really saw that the titers of this anti-HERV-K antibody really spike on therapy. So the titers of this antibody of interest are in red. And then we checked a different anti-ERV antibody as a control in blue. And you can see that only the levels of that anti-HERV-K antibody are spiking on treatment, which is represented by the gray shading. Now, this is kind of too modest of a cohort to jump to too many conclusions, but it's something we want to expand and understand if the level of increase in titer is associated with response. And also our collaborators in South Korea at the Samsung Medical Center have a really unique cohort of adenocarcinoma patients. All of these patients received immunotherapy. And while we couldn't check the patient plasma, this data wasn't available, we were able to look at RNA sequencing. So our collaborators looked at the level of ERV-K7 expression from their RNA sequencing data and correlated that with patient response to immune checkpoint blockade. And they could find that in patients that responded, they had a higher level expression of this endogenous retrovirus. And this also correlated with improved progression-free survival and overall survival in this cohort, even adjusting for multiple variables. So to summarize our B-cell paper, we found that a type of self-antigen, endogenous retroviruses, can elicit antibody responses, particularly in lung adenocarcinoma. The response to these antigens is boosted following immune checkpoint blockade. And this provides a mechanism behind correlations of B cells and tertiary lymphoid structures with improved responses to immunotherapy. But there are a lot of open questions that I'm interested in pursuing. For example, do we really understand where this maturation is occurring in the tertiary lymphoid structures or the lymph node? Does it matter to patient outcome? Are there other antibody responses to other potentially neoantigens or even non-epithelial antigens, for example, infections? And do those also help protect or improve the anti-tumor immune response? Do we see evidence of antibody responses over sites of metastasis? We really looked at the primary tumor, but we want to know what's happening over time. And then, of course, immunotherapy in lung cancer can also be given in settings of late-stage disease. I talked about early-stage resectable disease. And for the early-stage patients, pretty soon, these patients will also be eligible potentially for neoadjuvant immunotherapy. So it'd be interesting to understand this response and these different treatment settings. And then finally, B cells do not act alone. So understanding how the other cells are interacting with them to regulate, to promote or repress their responses will be interesting. And I think spatial technologies are key to addressing this type of question. So in the next few minutes, I'll just describe what work I did pursuing the spatial tumor microenvironment in London with the TraceRx study. So what we did here was we used a technology called imaging mass cytometry from our multi-region tumor samples. And we had a subset of the first 100 patients that had sufficient tissue remaining that we were able to put into tissue microarrays and then assess with this assay. Now, before in Vancouver, where I was doing, where we were using kind of 3 color stains, imaging mass cytometry allows multiplexing of 35 different antibodies. And it does this by, it's not a light-based imaging, it's the antibodies are tagged to unique metals. So it's a mass-based imaging approach, but you still retain that spatial information. So in TraceRx, we were able to acquire this imaging mass cytometry data, and we had, of course, the whole exome sequencing, RNA sequencing, and histology information available to kind of ask different interesting questions about the coevolution of the tumor microenvironment with the genome. So we have developed a method that I'll talk about now, but we have a paper that came out earlier this year. And we also have this available on GitHub for anyone who wants to use it. So this is just kind of walking you through the different steps. So we have our typical H&E that we're used to looking at and is routinely generated and clinically. And then we have our serial section that we did our imaging mass cytometry assay on. So here's just a pseudo-colored version showing different cell types and different stains. But this was very complex, and we had to go from this beautiful kind of staining images that we could put together, but we really wanted single cell data. So to get that single cell data, we started with a very specific nuclear segmentation. And when we started this project, we wanted to use machine learning, but there was no such trained model that we could use out of the box available in the literature for nuclear segmentation and imaging mass cytometry. So during the pandemic, we expertly labeled over 40,000 different nuclei from different histologies and different patterns of cellular organization. And this was used to improve our model for a very accurate nuclear segmentation. And this model is also available. We've tested it not only on humans, patient samples, but also on mouse models, and it works really well. So if anyone's using IMC and would like to use it, please let us know or follow that link. From those nuclear seeds, we then progressed to cell segmentation. This is just kind of used packaged in Cell Profiler, but we also have just an automated version that you can just run out of the box that uses pixel dilation around the nuclei. And then we have developed an automated cell phenotyping, which is a separate module that you can use from this TraceRx Flex pipeline. And we've shown that it works not only with imaging mass cytometry, but also with antibody and fluorescent-based technologies like PhenoCycler, Codex, and stuff like that. So it'd be interesting to hear if anyone wants to try it out on their technology. We also did tissue segmentation because we wanted to understand when immune cells were stuck in the stroma versus infiltrating into the tumor nest and ask various biological questions. This is an overview of the different markers that we used and the different cell types that we're able to resolve. So it was kind of a pan-immune panel. We looked at different T cell subtypes, different B cell subtypes, myeloid cells like macrophages and neutrophils. We had our tumor/epithelial cell marker, different stroma, so blood vessels and cancer-associated fibroblasts, and various phenotypic markers, including some checkpoints and things like proliferation and so on. And from this data set, we had over 2 million segmented cells. 29 different immune cell subtypes, tumor and normal epithelial cells, and 3 different stromal cell subtypes. I would say that another output from our pipeline is this pseudo H&E. So we worked very closely with pathologists on this study, and they're used to using the H&Es. So we used our best approximation of that from the IMC data for the pseudo H&Es, which was very helpful for getting their help for manual labeling of different structures. For example, we only had a pan-epithelial marker. So to define tumor epithelium from normal epithelium, they had to do these manual masks. And similarly, to differentiate between cancer-associated fibroblasts from large vessels, they also helped us with this, and from differentiating alveolar macrophages, which are a bit biologically different from like tissue-resident macrophages, for example. And of course, we were very excited to use the results of this to study different levels of spatial organization in the non-small cell lung cancer microenvironment. So this is going from single cells. So we have our tumor cells in red and various immune cells in different colors. We can then kind of group these into cellular communities. So these are looking for recurrently colocalizing cell types. And you can see that they kind of recapitulate different structures. They retain some sort part of the tumor architecture, even at the community level. And then finally, we had our broad TME classes, which used that tumor versus stroma tissue segmentation. And I won't really talk about those today. But then we could use the spatial data to ask questions about how they're related to different genomic features, for example, neoantigen burden, immune escape mechanisms, and of course, clinical outcome. And I won't go over many of the results today. This data is published, and you can always look up the other stories that we talk about after. So I mentioned that we have these cellular communities. We defined 10 different spatial communities in our study, and they're named by the cell types that are statistically enriched in them compared to the other communities. So you can see there's different tumor cell ones. So we have tumor border, tumor bulk, kind of coming up here, and lots of different immune cell subtype communities as well. And this is a published method. Now, we wanted to ask whether the formation of these communities harboring immune cells was related to neoantigen burden. And we were interested in clonal neoantigen burden because this is a genomic feature that has been associated with improved outcomes in immunotherapy-treated lung cancer patients and other cancer types as well. So we found that we did observe different histology-specific findings with associations with the burden of expressed clonal neoantigens. So in adenocarcinoma, we found that these communities of macrophages and T cells were associated with clonal neoantigen burden. This is just a zoom in crop of one such example. So we have the community in light orange and then the different cell types underneath. So we can see there are close interactions between macrophages, CD8 T cells in green and CD4 T cells in blue. And in literature, this proximity of macrophages and CD8 T cells has been reported to reduce their effector function and motility, and therefore may be a mechanism of extrinsic immune escape. So I think it would be interesting to know whether disruption of these communities would improve response to immunotherapy, for example. And we saw something else in LUSC, in squamous cell carcinoma. So we found that these communities of B cells and plasma cells was associated with clonal neoantigen burden. So this is one example. We have our community in purple, and then the cell subtypes beneath here. So we can see that most of the cells are plasma cells. That these cells were more common overall in the data that we had compared to B cells. But you can also see there are CD8 and CD4 T cells in there as well. So of course, we wanted to know, are these communities tertiary lymphoid structures, or are they distinct from that? And I think largely because we were working with tissue microarrays, excuse me, and we didn't capture any tertiary lymphoid structures in the study. So we can confirm that these kind of clusters of B cells and plasma cells are distinct from what you'd see if it were a tertiary lymphoid structure on H&E with these defined dark and light zones. But we're interested to know particularly because of these histology-specific differences that we've seen in the B cell endogenous retrovirus study, and also here, whether the role of B cells and plasma cells is different in these different histologies, or whether they're playing a similar role. So to summarize this section, we developed an experimental and computational pipeline for spatial TME analysis. And if anyone's interested in testing our pipeline, I'd be very happy to chat. We see that multicellular communities may organize in response to genomic aberrations in a histology-specific manner and represent potential targets to boost immune responses to clonal neoantigens. And while TLS were not detectable in the tissue microarrays that we used in the spatial study, further investigation into the role of these cell types outside of TLS will be interesting in the different histologies as well. And with that, I'd like to thank everyone that I worked with very closely on these projects. So in London, the Swanton Lab, Mihaela, Ali, Emma, and Claudia, various different collaborating labs across the CRUK Lung Cancer Center, and at the Crick, particularly Kevin Ng as well, who's now at Rockefeller. Our collaborations back in my PhD at BCCRI, VGH, and in Victoria, the funders, and of course, thank you so much for your attention as well.
Great, thank you very much, Katey. Is there, I guess we have some time for questions, so I'll open it up. If you just want to put your hand up in the on Zoom here, we can be preferable if you can ask your question. If not, add it to the chat and I'll repeat. If not, I can get started. This kind of these immunological differences between the two lung cancer subtypes is quite interesting. So, do you have any mechanism, especially for the self-antigen expression, that why it's specific to lung adeno? And you see quite a bit of diversity there, and obviously the response rates to immunotherapy is a lot different. I'm just wondering if you have any mechanisms that can kind of relate to that, or thoughts?
It's all speculative, but you've heard of people talk about B regs. So you can have these B cells that are actually immune suppressive. So it's possible that the phenotypes of these cells are just completely different in different circumstances, different histologies, but we didn't have the phenotypic depth to ask those questions. I think you need to do a different technology or design a specific panel to really look at that.
Yeah, and I guess there's no real lung squamous model that's very good for mouse models as well. And I guess since you started with that model, I guess you were kind of more inclined to look for things that were in lung adeno, I'd imagine, right? Because that's where your starting point was.
We were actually unbiased in the human data. If we saw something in squamous, I was kind of surprised that it was the antibody titers were specific to adeno because we saw expression in both.
Right.
So yeah, that's a bit of a mystery. And we also only did the, in that small group of 7 patients that had adenocarcinoma and were treated with immune checkpoint inhibitors. And we saw those spiking levels. We actually didn't look yet in squamous. So we have to still check whether adenocarcinoma specific phenomenon or if it's happening in other subtypes too.
Perfect. Great. All right, I'll open up. Spencer, you want to go ahead?
Hey, Katey. Thanks a lot for the talk. I was really interested in your spatial analysis, and I was wondering if you observed any increase in CD4 T cells with interacting with B cells in cases where you had the increased antibody titers. So like because of course when the B cells interact with T cells, that causes class switching, and I was wondering if there's any association there.
Yeah, I think, would you expect to see that because we didn't have any tertiary lymphoid structures in those images? I wonder if you'd still induce that class switching like anywhere kind of in the tumor microenvironment. But to be honest, we didn't check that by pairing the imaging mass cytometry data with that other study yet. So I could definitely look that up. That'd be really interesting.
Great. Kevin, you want to go ahead?
Yeah, sure. Great talk, Katey. So we've done something just kind of touched on looking at different cancer-associated fibroblast subsets in patient samples. And we had a really, really hard time with all of the heterogeneity, not just within the single section, but then between patients as well. And it eventually, I mean, we'll certainly pick your brain on this, but do you have a rule of thumb or a minimum number of patients or minimum number of TMA sections that you require in order to be comfortable making some of these conclusions? Because we were really struggled to look for any sorts of associations, because the heterogeneity, not just within, we had whole face sections, which was an advantage, but to just identify the right regions of interest to actually reflect what's going on in the tumor. But then as soon as you go to the next patient, it's just completely different. So, and head and neck is a lot more diverse. So that's probably one of the problems. But what's your kind of rule of thumb for these are enough patients, this is actually what's going on in this cohort?
It's a really tricky one. Because yeah, we did struggle with power, statistical power. So we kind of treated it a bit as a discovery tool, but we were stringent in the statistics because I guess our study was different because we had sampled different regions from a single tumor. So even though you had maybe a lower number of individual patients, you still had quite a high number of total area, if that makes sense. So we just used different statistical methods to correct for patient effects in all of our associations. But yeah, I think, there's the IMC study in lung cancer that looked at cancer-associated fibroblasts subtypes and found all those prognostic associations. I think they had hundreds and hundreds of patients, right? So it's definitely, it's tricky to know when you can be confident in something. And yeah, I don't have a rule of thumb particularly. I think it's
I think one of the advantages to some of your stuff is you're comparing to normal in some cases, right? If you're doing a tissue analysis, and that's certainly something that we did not do. But we really did struggle with that. But we didn't have hundreds of patients either. So we had, but we're looking forward to having some more thorough conversations with you about this. We can show you some of the data, and I think it'd be really helpful to us if we can get your eyes on it, because you've done this sort of thing a lot more than we have.
Yeah, that would be great. Yeah.
All right, we have a question in the chat. I don't know if you want me to read it, Katey, or do you want to read it directly? What's easier?
Sure, I can read it. So,
Sure.
Excuse me. Calves in the tumor stroma are likely to be influenced by being surrounded by macrophages to become more inflammatory, which can then have more crosstalk with immune cells. Did you have any cytokine or cytokine receptor in your immune panels? It's been shown that there's a correlation between inflammatory calves and worse patient outcomes. We didn't have single cell level cytokine information, but we just had kind of the paired region that had RNA sequencing. So we didn't go too deep into that actually in the calf angle, but it's something we could look at. We could look at specific cytokines. And then there's someone in the lab still in London who's quite interested in the calf story, and we have expanded the cohort, so that should be something she should look at. Yeah.
All right, is there any other questions?
Kevin?
Oh, one more for Kevin again.
Sorry, this one's a little bit more about a little less about our work and more about your work. So you've got a number of people in the pathology department at UBC here on this call. So what do you need to succeed? As a brand new PI, you've been here for all of 6 days.
Yes.
What are you looking for in terms of near-term, long-term collaborations, and what sorts of things are you planning to get these mouse models up and running? Do you need patient samples? What are you looking for?
I'm really interested in learning more about the patient samples and biobanks that are available. I'm really interested in the lymph node and what's happening in patient lymph nodes, which I know is very challenging to sample. So I think I'd really love to talk to more like pathologists and maybe lung cancer surgeons to understand the limitations and what's possible there. But I think I'm quite interested in studying what's happening in the human samples, maybe moving to something like spatial transcriptomics, or even collaborations with, there's someone in Boston who can do spatial BCR sequencing. So having those kind of collaborations and setting up technology that could do that would be really cool. Yeah.
Sounds good. Are you willing to get out of lung cancer? Because there's some other tumor sites where they resect lymph nodes on a regular basis, and getting tissue is actually not an issue.
Okay. Yeah, definitely. I mean,
Depends on the
these
tumor site.
questions- I mean, all tumors, well, if they're a certain stage, they've spread to the lymph node. So I wonder, what's common across cancer types in that problem.
Stop trying to pull her away from lung, Kevin. Okay, any other questions?
Guys, it's
All right.
John English here. I'm compelled to just make a statement about the lymph nodes. So in support of our pulmonary database, we certainly have many lymph nodes that come out with the resections for lung cancer. So stay in lung, Katey.
Okay.
Thanks. Good talk, by the way. Great. Nice to see you back. Nice to see you back.
We'll catch up
Thanks,
soon.
John. It's awesome. Okay. Well, if that's all for questions, we'll thank Katey one more time and welcome her to UBC, or again back to UBC. So, and look forward to seeing you around and what you do in the next couple of years. It'd be great. Awesome.
Thanks, everyone. Yeah, good to see you all. Talk to you soon.